tack that I'm going to take with my particular talk is discussing how malaria can be thought of as a re-emerging infectious disease because of the issue of drug resistance. And this is something that Chris mentioned and gave a great introduction, actually. So I don't think I really need to go through my first introductory slides, but just to say that the malaria parasite, is resistant to almost all antimalarial drugs that have been developed, and as well as the mosquito vector being resistant to insecticides as well. So what sort of, how does this fit into controlling malaria, which Chris also defined too? Well, we basically have two methods for controlling malaria. The first is to control the malaria parasite, and this is through the use of antimalarial drugs, but also malaria vaccine, which was also discussed a little bit, which has not been very effective at all and is not widely available, or to control the mosquito vector through the use of insecticides and bed nets, also personal protection methods, for example, some of these coils that I'm sure some of you have used, or for example, draining bodies of water. So I'm really going to be focusing on controlling the malaria parasite through the use of antimalarial drugs. And what are some of those drugs? Well, some of them are listed here. Perhaps I can just have a show of hands, actually. How many people in the audience have taken an antimalarial drug? So quite a number of you. Does anybody recognize these drugs? Yeah, okay, so I'm seeing a few nodding heads here. Different levels of efficacy, but resistance has basically arisen to almost all of them. For treatment, then, for Plasmodium vivax, ovale, and malariae is chloroquine, and then add primaquine for Plasmodium vivax and ovale because of the dormant liver stage that they have. For Plasmodium falciparum, which has become resistant to chloroquine right from the early 60s, we use artemisinin and combination therapy, ACT, and then also primaquine to get rid of the sexual stages of the parasite, and then intravenous quinine for severe malaria as well. So as I mentioned, the problem is that malaria parasite has become resistant to almost every drug that has been thrown in its path. The resistance has arisen in several foci. This is a diagram, schematic that I generated. You can see the endemic areas are in light blue here. Chloroquine resistance is indicated in slightly darker blue, and then artemisinin combination ACT resistance is indicated in dark blue. And the spread of resistance is indicated by the arrows. So here you can see chloroquine resistance arose in several foci in different countries, but then spread, as did pyrimethamine and sulfadoxine. They're usually used in combination, these drugs, through migration as well. And then the most serious, most recent cause for concern has been the rise of this PF Palin strain, which is resistant not only to artemisinin combination therapy, but also to the, or artemisinin I should say, but also to the combination drug, which is also used for the treatment. So now that we have this level of drug resistance that's being spread throughout the world, what can we do about it and how can we monitor the situation? So there has been a lot of research over the past 30 odd years or so to identify molecular markers of resistance, and examples of these are listed on the left-hand side. So this is the gene, and this is the particular mutation in the codon which causes an amino acid change and enables the parasite to efflux the drug or in some way to be resistant to the drug. So you can see that we have almost 6 or 8 genes now, genetic markers, which can be used for surveillance studies. An example here is a very classic example. Really one of the first molecular markers was the PF chloroquine resistance transporter gene, PFCRT. This is a schematic of the protein you can see here. It has at least 8 transmembrane domains, very complex genetic structure, and mutations, in particular this K76T mutation here, causes or enables the protein to export chloroquine from the parasite back out so that it becomes resistant. So the molecular markers that have been developed then, we have started to use in our studies, and I'm going to tell you a little bit about our work in India. Chris alluded to this as well. NIH has funded these International Centers of Excellence for Malaria Research, or ICEMERs, back in 2010. There are actually a total of 11 of them, all over the malarious world. You can see here, these are the countries which are represented by the ICEMERs. Our particular ICEMER is based in India. It's called the Center for the Study of Complex Malaria in India. And these are very large regional centers supposed to set up a network of centers across the malarious world. They're 7-year grants. They've just been renewed. We all have to study the epidemiology, vector, pathogenesis of malaria in our particular countries. We have to be responsive to changing epidemiology situation. There's a big focus on capacity building, which is actually quite a change for NIH. And as directors, both Chris and I are directors of our respective ICEMERs, we have to spend at least 30% effort in the countries that we do our research in. And we're overseen by a scientific advisory board as well. I should say there's a direct relationship between the ICEMERs and the in-country control programs too. So a little bit about malaria in India. India is a fascinating country. It's the largest democracy, over 1 billion people. It's topographically and climatically very diverse. Malaria, there are actually 2 species, Falciparum and Vivax, which I mentioned previously, which are very predominant, and at least 6 mosquito species which transmit the parasite. This is a map of India here, and this is what's called the annual parasite incidence, which is basically an indication of a large malaria prevalence. So you can see, in particular, in the northeastern part the eastern part of the country, and then the northeastern states here, connected through what's called the chicken neck up here. Malaria in India has declined over the past few years with a few increases, but actually 2017, 2018 are not shown here, but there's also been a massive decrease. And India has actually entered malaria elimination phase as well. So our particular field sites then are based at 6 different areas in India, in particular in parts where there's a high IPA shown here. And I just wanted to, see if this works, take you to the field sites. This is a Google Earth map. It always irritates me that it starts on the US. I don't know why it does that. We're moving over to India here, zooming in to one of our field sites, which is in Odisha. So this is a very forested area. Obviously there's been deforestation in certain parts of the state. But you can see here, this is the main town, Rourkela, where we have Ispat General Hospital, which is where our main collaborators are, and Dr. Sam Wasmer is actually in the audience today, who's come from the London School of Hygiene and Tropical Medicine for this symposium. And you can see here some of the villages that our epidemiology teams go out to. So this is a more forested area. This is actually one of the villages close to a forest where you get, in particular, a lot more malaria cases. Other villages which are in areas where there's been deforestation, there is less malaria. I think I'm going to leave that there actually and not show you the other sites. But just to show you perhaps some of the terrain as well. Chris mentioned this, malaria is a disease of poverty. We routinely, let's see if this will come up to, travel to one of our field sites again by train. This is a video taken outside from the train. You can see groups of people outside enjoying football matches, cricket matches, very lush. This is actually during the rainy season. You can see some of the houses. Animals are living quite close to the houses in some cases. We're just coming up to, you can see overhead dark clouds. It's the rainy season. And these are paddy fields, so these are prime Anopheles breeding sites. So that gives you some idea of the ecology. So what we've been doing then at these different field sites is renovating, staffing, equipping the sites, training, so a lot of capacity building. These are some of our field teams in Shillong and then also in Rourkela. And we've set up several epidemiology and vector surveillance projects since about 2012. Have enrolled more than 15,000 subjects. And all of these, first off, have a census in the particular household where we visit, and they can either be cross-sectional, where we go out into the community, longitudinal, where we also enroll people and then follow them during time, or passive surveillance, where people come to us, to the clinic that has been set up too. And and we also do adult and larval collections as well at these particular villages. And for each subject who's been consented and enrolled, samples are collected. So we do a finger prick for a small blood volume, a blood smear, a rapid diagnostic test, a blood spot. And if they're positive, we take a vacutainer. And then all of these samples get stored appropriately for downstream assays. Everything is collected into a REDCap database as well, shown here. And we have very detailed clinical questionnaires to help us too. So these samples then are the samples that we have started to test for drug resistance, obviously to get some idea of how drug resistance has been spreading about India. And the way that we've done that in particular is through development of a next generation sequencing core in Delhi. This is part of our capacity building. So we set up what's called an ion torrent machine, which is a high-throughput sequencer. We had a couple of postdocs, Pavitra Rao and Swapna Appelko, who were basically providing support 24/7, purchasing and shipping of next-generation sequencing kits from the US to India because they weren't always available there. And we developed a high-throughput genomics assay to survey these drug resistance mutations I was telling you about. So in particular, we've been able to now rapidly identify drug resistance mutations in 6 complete genes, not just partial parts of the genes, in, for example, 96 samples at a time. And this has enabled us to identify several interesting things. For example, this is the artemisinin gene mutations in which cause artemisinin resistance, the CALCH13, and we've identified new mutations there not actually associated with drug resistance. And then also at the same time, you can see this is one particular patient who's had his or her parasites genotyped at these 3 different loci, and you can see that there are mixtures. So half of the parasites are resistant, half of them are sensitive. So we can actually identify resistant and sensitive clones in the same patient. So this is very much an early indicator of sort of drug resistance emergence as well, and that was published a couple of years ago. More recently, we've scaled up and done high-throughput analysis of actually close to 200 falciparum samples. You might actually think that that's quite a small number. In fact, it's really already revealing patterns and some reversion to sensitive type, which is actually a phenomenon that Chris first identified in Mali several years ago. So you can see here, this is a map of where the 3 particular sites where we've been taking samples from, and these are the results on the right, shown in pie chart form, with resistant being the resistant form of this particular gene, the chloroquine resistance gene in this case, and blue and green being the sensitive. So you can see that we still very much have chloroquine resistance alleles here present in Chennai and Nadiad, but actually in Rourkela there seems to have been reversion to sensitive. And this is very important because it may mean that we can start to reuse that drug at that particular site. The drug pressure has been lifted, the parasite has returned to sensitivity, and there's a possibility we could start to reuse that drug. This is another gene, the PFMDR1 gene, and there are resistant mutations which vary in frequency at all 3 sites. This is the PFDHFR and PFDHPS, which are involved in pyrimethamine sulfadoxine, that drug combination I told you about, also with different what are called haplotypes. So far we haven't found any of the artemisinin resistance gene mutations. These are the 2 new mutations that we identified here. However, unfortunately, just in November last year, there was the first publication in the New England Journal of Medicine of artemisinin-resistant falciparum in West Bengal, which is shown here. And if you can see, West Bengal actually sits right on top of one of our field sites. So we are ramping up now to screen the rest of our samples to try and see if we have any evidence as well for artemisinin resistance. One final thing I should say is that we've also adapted the high-throughput genomics assay to a portable handheld device. The samples from the field were being shipped to Delhi to the genomics core there, and that caused delays, and it wasn't real time, and sample loss as well. And so we've been working with Oxford Nanopore Technologies to use this handheld device. And basically the DNA strand moves through a pore in a membrane, a flow cell, and whether it's a G, an A, a T, or a C is indicated by a change in potential. You just hook up this device to a laptop, so it's very easy and very portable too. So we've adapted our particular high-throughput assay for use on this handheld device, and now we're taking it directly to the field to identify potentially drug-resistant parasites more in real time. Okay, I'm going to leave it there. So a summary, malaria parasites have developed drug resistance to almost all anti-malarial drugs, really since World War II when we first started developing them. Re-emergence of malaria due to spread of anti-malarial drug resistance is an enormous concern as we enter the elimination phase in many countries and in India too. And introducing high-throughput technologies directly to field stations enables more real-time surveillance and prevents problems of sample loss during shipping or delayed analysis. A strong surveillance system is key to monitoring and containing the spread of drug resistance as well. And this appearance of mutations associated with artemisinin in India is of high concern. Okay, I just want to say thank you then to many of my collaborators at the National Institute for Malaria Research, the Carlton lab, the NYU Genomics core here, which is a fantastic facility that Elodie now oversees, as well as people at the Nanopore company as well, where we've been working hand in hand with them. So thank you.